Okay, welcome back. Hope you had a good lunch. And is this echoing? Is this okay? Okay. So I know it's very hard for people listening because it's like drinking water from a fire hose. You know, getting all this stuff going very, very high pressure, and I know it's hard sometimes to keep track. So if I'm going too fast or there's a concept you want me to repeat, just raise your hand, don't be ashamed, and I'll go over it again. And also, I'm from New York City, so people from New York tend to speak very quickly, because if you don't speak quickly, then, you know, they're gone, whoever you're speaking to. So again, if I'm speaking too quickly, just tell me to slow down, okay? Any questions or anything you want me to change or do differently, tell me. Don't be, yes?
One question. You talked about how white cells can find their way out of the circulation at the site of inflammation, and these adhesion molecules play a role in directing them. The selectins, are they constitutionally expressed on epithelia, or are they upregulated as well? Because they appear to play an important role in sort of slowing down the cells and maintaining the cells in the periphery so they can access the endothelial cells.
Yeah, they're expressed at low levels continuously. But then what happens with a lot of these factors is that when there's activation and inflammation, they can get raised to even higher levels of expression. Okay, so now things will start getting interesting, because now we're getting into T cells and B cells, and also, unfortunately, it can get more complicated. So the third lecture is going to be discussing antigen recognition by T lymphocytes, and the third and fourth lectures are really partner lectures, because the third lecture, conceptually, is going to be looking at what goes on outside of the cell, how T cells are seeing antigen, and the second lecture part, which is lecture 4, will be discussing the structural motifs and how things go on inside the cell in terms of presenting the peptides. So again, it's good they're partner lectures. So to start off with, the questions we want to consider are, first of all, how do B cells and T cells see antigen? And how does a T cell distinguish between whether a foreign antigen is being presented to recruit help or because the cell is infected? And obviously, if it's to recruit help, you don't want to kill the cell, you want to help the cell. And if it's because the cell is terminally infected and can't be saved, then you want to kill the cell. So how do T cells know how to distinguish between those scenarios? Why do some T cells kill and others provide help? Now, clearly there's a psychological explanation for that, but as immunologists, we have to come up with more of a structural understanding, a molecular understanding of why that occurs. And so again, these are the things we're going to talk about in this particular lecture. Now, this is actually one of my favorite slides, because this is really what immunology was about, like maybe 25 to 30 years ago, in terms of its understanding of what was going on in the process. But the other reason I find it fascinating is it really talks about how when you don't know something, you trivialize it and say that it's not very important. And then all of a sudden you start realizing that something that wasn't important turns out to be the critical link or most important aspect. And this turned out to be the case for macrophages and monocyte-derived macrophages, because about 25, 30 years ago, we basically thought that these cells were just phagocytic cells. All they did, they were the garbage people of the immune system. They circulated around, they saw something that needed to be taken up, garbage picked up, they just swallowed it, they chewed it up, and in essence that's what happened. So you have this phagocytic cell, it sees a bacterium, that's what they called it back then, and then binds to it. You have some membrane ruffling. They were very proud they knew about membrane ruffling back then. It ingests it. It brings it inside. You have fusion with the phagolysosomes and digestion, and then there's this kind of non-specific burping process that occurs when this is just kind of like shed out into the environment. And so in essence, macrophages were really looked upon as, just non-important cells. And the really smart people in immunology didn't waste their time studying macrophages. They were really more interested in other very more complex cells. But now it turns out, and as this lecture will show, macrophages are really clearly the critical cell in the process of T cell and B cell immunity. And these cells, now, whenever you want to raise the importance of something, you give it a title. So instead of calling it a phagocytic cell, now you got a promotion, and now it's called an antigen-presenting cell. And that sounds a lot more sophisticated. And in New York, what they did was we had people who were working taking out the garbage, and they were called garbage men. And they wanted to get a raise, but the city was bankrupt. So instead of giving them a raise, they called them sanitation engineers. And they were very happy with that. They went back to work at the same salary. So pretty much the same thing happened with these macrophages. They really wanted to get something more important, and therefore we renamed the antigen-presenting cells, because that's teaching us that their role is not so much ingesting, which also it does do, but in terms of presenting antigen, particularly to T cells. And the two major players I'll discuss initially are macrophages and dendritic cells. And macrophages are critical phagocytic cells, and in fact, they have an important effector function in terms of their ability to phagocytose and kill bacteria, and that plays a critical role in the immune system. As a secondary process, they also are involved in antigen presentation. Dendritic cells, they have phagocytic capabilities, but as opposed to macrophages where that phagocytic capacity is being used as an effector function, but dendritic cells primarily, their uptake of antigen plays a critical role in terms of enhancing its capacity to present antigen. And again, as the lecture goes on, I'll discuss distinctions between macrophages and dendritic cells in terms of their function. Now, if we think in terms of infection with TB, which is something that several of you are involved with, we have two possible scenarios. The first scenario is that you basically have the phagocytic cell. It sees the Mycobacteria tuberculae in circulation. It phagocytoses it, and that's the end of the story. So it now has the MTB sitting in the vacuole, but apparently it's not yet fully activated, and if it's not yet fully activated, it lacks the ability to destroy the TB inside the vacuole, and this is what you would see now in this particular scenario. People who are in the front could see large numbers of these classic acid-fast bacilli, which represent MTB. They haven't been destroyed, and therefore the infection continues very strongly, and ultimately this will lead to significant disease. In contrast, if this macrophage is activated by virtue of interaction with the T helper cell, this T helper cell secretes factors. We'll discuss in a few minutes. Gamma interferon is a critical factor produced by T helper cells. This now activates the macrophage, makes it a much more effective killing cell, and now these MTB that previously have been able to replicate inside these vacuoles and then ultimately escape into the cytosol, now because you upregulate the phagolysosomes, they merge and fuse with the vacuoles and now kill the TB, and now in this scenario, the infection is being cleared. So clearly, the macrophages play a critical role in terms of its ability to control HIV infection, but in order for that to occur, it has to be an activated macrophage. A resting macrophage just does not have the digestive juices going enough to kill TB, and what provides the stimulus for that is the T helper cell. So this is this interplay that you need between macrophages and T helper cells. And after a macrophage is activated, again, it has large numbers of these phagolysosomes able to fuse with the vacuole. Also, it upregulates a whole host of surface molecules, so it will upregulate MHC class 1, MHC class 2, which as I'll discuss later, makes it much more efficient antigen-presenting cells. It upregulates molecules such as B7 and CD40. I'm going to discuss in a later lecture that play a critical role in co-stimulating T-cell activation. Tumor necrosis factor, as you know, plays a critical role in terms of vascular permeability and cellular migration. And nitrous oxide plays a critical role also in terms of killing. This is an activated macrophage. And again, the immune system has the same motif and approach that I previously spoke about with the lymphocytes. If there's no infection, cells are not activated. So if there's no active infection going on, there's no need to waste all the energy to have this macrophage making all the proteins and all the factors that are required, and therefore it's basically relatively quiescent. But the trade-off of that is that in case infection occurs, it takes a while to mobilize. So it's kind of like you turn on a computer till the hard drive kicks in, till everything kicks in. It takes a while to occur, and during that window of time, clearly the infection can go on uncontrolled. Now, this is, again, you're going to see this slide a couple of times, and just like this slide illustrates the secondary response. So every time you're going to see this slide, you can develop a more rapid response to seeing this slide. But just again to illustrate, the first time you see antigen, there's a lag phase of about a week before you get any response. You get a slow response. It's not very high. However, the second time you see antigen, you have this very dramatic, rapid response within a day or two. And again, the level of the response is dramatically better. So what this allows us to really identify also is whether you've seen antigen and what the response has been. Now, if we look in terms of the T-cell macrophage interaction, so here we have an infected macrophage. It's been infected with TB present in the vacuole. It's not been activated enough, so it's not able to efficiently kill it. Now the T helper cell comes along. It recognizes that the macrophage is infected because in this case, as we'll discuss in greater detail, it's seeing a peptide derived from the TB in the MHC class II molecule, and now it, again, showing co-stimulation with CD40 ligand. Again, I'll discuss that in greater detail later. But just to focus now, it's making large amounts of gamma interferon, and the presence of gamma interferon activates the macrophage to make it a more effective killing cell. So therefore, these are the major players now that are playing a role in TB immunity. Now, this is another thought experiment for you to do, but now you actually have graduated to using mice for your experiments. So with that information that I gave you, here's an innocent mouse, look in those eyes, just staring at you, saying, please make the right decision to save my life. And the question would be, transfer of which cells would protect this mouse from TB? And again, get ready to vote, because I'm going to ask you to vote. And the choices basically are the following. Either you could transfer activated monocytes from another infected mouse. So you infect another mouse, you activate the monocytes, they're now revved up to kill, and now you can give, for example, say 1 million cells transfer intravenously into this mouse. Or you could transfer T cells derived from an infected mouse that has undergone the secondary, that now is undergoing a secondary response to this mouse. Or you could transfer B cells from a mouse that also has been exposed to TB and undergone a secondary response. So you have one of 3 choices, and you cannot choose all 3, okay? So the first, who votes you should use activated monocytes to protect this mouse? Raise your hand. Now again, if you raise your hand, go for it, okay? You know, no halfway votes. So who would go with activated monocytes? Raise your hand. Okay, makes sense. They're the ones that are the effector cells. Who would go with T cells? Raise your hand. Okay, well, T cells are always a safe answer because they're important cells. That's a good one. And again, you know, clearly they're playing a role. Who would go with B cells? Raise your hand. Well, that's a smart group because I haven't even talked about B cells, so how possibly could that be the answer? And it's like any multiple choice. You always throw a third choice in just kind of out of the blue, you know, because you have to fill it up. If you had 2 choices, it would be a pretty easy question. So now we have to ask the question between activated monocytes and T cells. It turns out that if you give the mouse activated monocytes, it really doesn't help very much. The mouse actually dies from TB infection. However, if you provide it with T cells from a mouse that has previously seen TB, that actually saves the mouse's life. And the question is, why should that be? And the answer basically is that as I showed before, an infected macrophage, if it's not activated, it won't be able to kill. However, once the T cell activates it, now it can efficiently kill. When you're transferring over, say, 1 million activated macrophages, in the greater scheme of things, it's not a lot of cells. So therefore, that's not enough to turn the tide against TB. You may have to give, who knows, 100 million activated macrophages. However, when you transfer the T cells, these T cells now recognize the TB peptide being presented to it, so a single helper cell could go on to activate thousands and thousands of macrophages. So you have a tremendous potential for amplifying the immune response, and that turns out why T cells really are the cells that you'd want to transfer into that, particularly CD4 T cells, into that mouse to save that mouse's life. So that's why that answer works. But again, doing that experiment allows us to really appreciate the capacity of T helper cells to rapidly mobilize and activate macrophages to make them be able to effectively kill it. So in essence, the T helper cell, think about as a capacity building cell. Because what it's doing is it's coming into the environment and building the capacity of the macrophage to be able to eliminate TB. And again, the reason that's also important is if you'd be designing a vaccine, the kind of vaccine you want to design is one that's going to activate T helper cells that can now turn on macrophages and make them able to eliminate TB. Okay, any questions? Okay, now, well, how does the T cell know that the macrophage is infected with TB? Again, it's not like T cells are mind readers. They're able to just guess by the way that the macrophage is diapodecing, that it looks like it's an infected molecule. And it turns out it's going to have to do with the way that antigen is being presented to the T cell. Now, this basically illustrates how B cells and T cells see antigen. As I had mentioned before, they use different antigen-specific receptors. This is an antibody molecule. Again, if you remember, the heavy chain, light chain, the combination of the heavy and light chain is providing antigen binding site. The immunoglobulin molecule, in this case, for example, IgG, would have two binding sites, one here and one here. This is the T cell receptor, same concept, constant region, and then a small hypervariable region that's recognizing antigen. And again, this structure and these are completely different. However, if you look at this picture, what jumps out as a difference between the immunoglobulin molecule and the T cell receptor? What do you see that's different? There's two of them, two immunoglobulin and one T cell receptor. Why are there two of them? What's the difference between this one and this one? One's attached and one is not attached, whereas the T cell receptor is only attached. What is that teaching us? That's teaching us a very important difference between the antibodies and T cell receptors, because the antibody molecule has two functions. One function is at the cellular antigen receptor. So it's basically bound to the cell, and it's telling the B cell whether or not the antigen is pre-programmed to recognize, is in the environment, and hooked up to all the signal transduction pathways to activate that B cell and make antibodies. But in addition, the effector molecule for the B cell is the antibody molecule secreted by that B cell. So it basically secretes a antibody molecule that matches the one expressed on the surface, and this is the effector molecule of the B cell. So the B cells don't have to be anywhere near the infection site in order to be effective, because the antibodies are actually doing the clearing of the infection. However, the T cell receptor only exists bound to the cell. It only serves as the indicator to the T cell that the antigen it recognizes is out there. It activates the T cell, but the actual function is not carried out. There's no soluble T cell receptor to get into the circulation to look for a peptide. It now activates the T cell, which means that the T cells themselves have to get to where the infection is. They are like hand-to-hand combat type effector cells in the immune system, whereas one can think of the B cells as shooting missiles, and the antibody molecules are the missiles that they shoot, but they're very tightly guided by virtue of the antigen binding site that they recognize. Okay, is that clear? Now, in terms of the receptor gene, I'm going to discuss the molecular rearrangement in a little bit more detail when I discuss the immunoglobulin molecule, but again, the concept is the same. If we would have to have a single gene for each possible T cell receptor, we would basically take up the entire genome with genes encoding T cell receptors and antibodies, which is obviously very, very inefficient. Because as important as immunologists think they are, we're not important enough to take up the entire genome. So what the immunoglobulin and T cell receptor gene has done is use this incredibly efficient way of basically picking and choosing and putting together different sequences to dramatically increase the capacity to generate diversity. And again, I'll discuss this in more detail with the immunoglobulin lecture, but all you need to know at this point is that this locus can have 70 to 80 different variable regions. Then there's J regions or joining regions, and each one of these has a completely different sequence. Each one of these has a completely different sequence, and by virtue of picking one of these and one of these, that gives you a completely unique sequence. And think about it as like on those luggage cases where you have those combination locks, where you have those 3 wheels that you turn, and 3 wheels don't take up a lot of space, but by having 0 through 9, you can get like, what, about almost 1,000 combinations. And imagine if instead of having just 9 numbers or 0 or 10 numbers on each one, you had 100 numbers on each one. Imagine how 3 wheels can give you that much high level of diversity that no one could ever randomly guess what it is. So too, by having all these multiple pieces, and how they combine together gives you tremendous diversity. The beta locus, in addition to having the variable region and the J region, has an additional region called D region. And again, the way that I always remember it is, again, friends of mine are from Brooklyn, and the way they would pronounce beta is beta, beta cells. So think of beta with the D, and that's how you remember that the beta locus has the D, and the alpha chain doesn't because there's no duh sound in alpha. So again, these things, try to help you remember these things. Okay, so now if you want to put it into context, now you have all these variable regions, all these J regions, and here you even have the additional variety of the D region, and now you basically pick one V, one J, and this unique combination now gives you a unique gene that can see something. So if you take this V and you bind it to this J, you get one sequence. If you take the same V and bind it to a different J, you get a different sequence which is going to recognize a different antigen, and so forth and so on. So by taking a single V and matching it with dozens of J's, you get tremendous diversity. And now that gives you an alpha chain. The beta not only has the diversity of the J, but also has all the diversities of the multiple Ds. And therefore now an alpha pairs with a beta. And again, the same way I mentioned with the heavy and light chain, the same alpha paired with a different beta will see different peptides. So by now having 2 chains, again, you dramatically increase the amount of variability. And again, I'll discuss this again in greater detail with the immunoglobulin rearrangement, but again, these are the same concept of this cassette approach of taking pieces of DNA, bringing them together, and then by having different pieces with different sequences, it generates a tremendous amount of diversity. So now, in order to understand how T cells and B cells see antigen, we take advantage of this secondary amplified response. Because the first time a B cell or T cell will see antigen or think it's seeing antigen, it will mount a low-level primary response. However, the second time it's seeing antigen, it will mount a more rapid, higher-level secondary response. And that's a way of now using to find out how the B cells and T cells are seeing antigen, because we'll change the antigen and see if the T cell or B cell recognizes it as a new antigen or as an old antigen it's seen before. So the experiment that we would utilize is by immunizing with, in this case, a native protein, and then we would boost with a native protein. Well, it's the same protein, right? So obviously, both the B cell response and the T cell response will provide you with a secondary response. So if we use this as a prop, so this is obviously a bag, and if I blow it up, this has a tertiary structure, right? It looks a certain way. But if I take the same bag and I basically flatten it out like that, it obviously has a completely different tertiary structure. So in essence, we could do the same thing with proteins. We have a native protein that has the normal configuration, but we denature it, flattened out, has a completely different appearance, and now we ask the question, do B cells and T cells see it as a different antigen from the native or as the same? So we immunize with the denatured protein, so we'll immunize with a flat paper bag, now we'll boost with the native protein, and lo and behold, the B cells do not develop a secondary immune response because to them it's a completely different antigen, whereas the T cells mount a perfectly good secondary immune response. If you immunize with the native protein, come back with denatured protein, again, B cells do not see it as a same protein, whereas the T cells say, yeah, it looks the same to me. And just to prove that this flattened denatured protein is immunogenic, if you immunize with denatured, come back with denatured, B cells work perfectly fine. See it as a secondary response, sees it as it's seen it before, and again, T cells are always seeing it the same. So clearly, this is teaching us an important lesson that B cells and T cells see antigen differently. And it turns out, as I alluded to initially, that it depends upon what exactly that they're seeing. And another important observation is that whereas B cells can see antigen that's free-floating, if you take free-floating antigen, like this paper bag, and add it to a T cell, the CD4 T cell will have absolutely no reaction at all. However, if you take this same bag and you mix in some macrophages, and now you mix it in with the CD4 T cells, as I'll discuss, what's now happening is that a peptide from the antigen presenting cell is being presented to the T cell, and now you get an immune response. So in essence, what's happening is the macrophage is taking this bag and it's basically ripping it apart into pieces of peptides, and that's actually what the T cell is seeing. So in the absence of the macrophage, it can't do anything with this bag. Only when this bag is digested into these little peptides, now is it able to see antigen. Is that clear? But the other important thing it's teaching us is why the T cell doesn't care what the structure of the protein is. Because it doesn't matter whether it's native form or whether it's the denatured form, at the end of the day, it's seeing the exact same peptide. So the structure of the protein is irrelevant because at the end of the day, it's only seeing a peptide, and that's why it mounts the same secondary response, whether or not it's seeing the antigen as a native form or in the denatured, because it ultimately gets digested up to the same peptide regardless of the structure, tertiary structure of the starting protein. Is that clear? And this is just a kind of a little bit more complicated experiment to basically show that while the antigen must be processed to peptides, still if you pre-digest it, it works just almost as well. So in this case, you basically take antigen, the antigen presenting cell, digest it into the peptides. It places the peptide into the MHC molecule. Again, I'll discuss this in a little bit more detail later in the lecture. But now it presents it to the T cell receptor, you get a response. If you take the same macrophage, it ingests the antigen, but you fix it with glutaraldehyde and therefore prevent the membrane from allowing any internal new peptides to be presented, you get no immune response. But if you fix the macrophage with glutaraldehyde so it has the MHC molecules, but now you artificially digest the protein in the constituent peptides, these peptides can lodge in the MHC molecule and be presented to the T cell. The bottom line is you need two things. You need to have peptide, and you also need to have an MHC molecule to present it. If you would add free peptides to the T cells, again, that would not be sufficient for the T cell to recognize. Okay, is that clear? And again, now to come in, how do B cells and T cells see antigen? So what a B cell is seeing, what an antibody molecule is seeing, is a three-dimensional structure of the protein. So it's very superficial. It just says, well, if it looks like this, that's one protein. If it looks like this, it's a completely different protein. And therefore, that's why when it sees denatured or native, it thinks of it as completely two different proteins. However, what the T cell is seeing is a peptide, and the same peptide is going to be derived whether or not the protein was wild type or denatured, and that's why it doesn't matter to the T cell what the primary structure of the antigen is, whereas to the B cell, that's incredibly important. So it's because it's recognizing a three-dimensional structure provided by the antigen structure. And the three-dimensional structure of native antigen is going to be different from the three-dimensional structure of a denatured antigen. Is that clear? Now, this is, again, one of the critical questions. Well, why don't you have self-reactive T cells, and are peptides that are being presented in the MHC molecule, are they only foreign peptides? And again, this is a very common misconception. When people first learn immunology, they think that only foreign peptides are presented by MHC molecules, and that's why we don't have any immune response targeted against self. It turns out to absolutely not be true. In fact, if you're not infected, all your MHC molecules contain self-peptides. because that's all there is out there. So there's pretty much no such thing as an empty MHC molecule. It always has to have a peptide in. If you're not infected, the only other source are self-peptides. So now you have self-peptides in your MHC molecules. Why don't you now mount an immune response against self? And the answer is because there are no T cells responsive to self-peptides in circulation because they've been removed in the thymic selection process. So even though you're expressing thousands and thousands of self-peptides, as long as there are no T cells to recognize them, you won't get an immune response, and therefore you won't target those cells. The flip side also is, early on in the infectious process, you could be presenting viral peptides, but if you haven't yet mounted an immune response, again, you won't be able to recognize them. So the MHC molecule has absolutely no idea whether the peptide is a self-peptide or whether it's a foreign peptide. It will display it, and it's depending upon the presence of the T cells that are recognizing in order to mount an immune response. And foreign peptides and self-peptides are processed exactly the same way. There's no, that I know of, advantageous processing of foreign peptides over self-peptides. It all comes down to whether you have these T cells in circulation that recognize it. Is that clear? It's a very important concept. So there are two kinds of MHC molecules. There's MHC class 1 and MHC class 2. And Dr. Janeway, in his book, showed it very clearly because MHC class 1 has 1 foot. So this is, I guess, like that person trying to learn all of Talmud while standing on 1 foot about Judaism. And MHC class 2 has 2. And again, so whenever you see the picture, it's very obvious which one he's referring to. But again, clearly they have more differences to their structure than just whether they have 1 or 2 feet. And they also turn out that different cells express different groups of MHC molecules. So in terms of MHC class 1, pretty much every nucleated cell in the body expresses MHC class 1 molecules. You name a cell that's nucleated, it will express class 1 MHC molecules. The only cells in the body that don't express it are red blood cells. And again, red blood cells are non-nucleated cells. However, when you look at MHC class 2 expression, it turns out to be very, very specialized only to what we would call antigen-presenting cells. And these include T cells, B cells. Again, as I mentioned, the important antigen-presenting cells are macrophages and dendritic cells. Epithelial cells I'll discuss when I talk about thymic maturation. However, all other cells in the body, neutrophils, you name them, they do not express class 2 MHC molecules. Obviously, red blood cells don't either. So what that's teaching us is the capacity to express MHC class 2 molecules is unique to immune cells, and that means that this is playing a very different role in terms of focusing an immune response than MHC class 1. MHC class 1 immune response is going to be focused in a way that every cell potentially can interact with a CD8 cell, which I'll discuss in more detail, because immune surveillance to eliminate infected cells is applicable to all cells in the body, whereas activating immune response recruiting help are only relevant to cells in the immune system that need to get that help in order to become better effector cells. Okay, is that clear? And in terms of, well, what cells recognize MHC class 1 versus class 2? As you all know, T cells come in 2 types. One group of T cells are CD4 cells, one group are CD8 cells, and they're defined by expression either of the CD4 molecule or the CD8 molecule. And this is just showing a ribbon structure of the molecules. What actually is kind of interesting is that CD8 actually comes as an alpha and beta heterodimer, whereas the CD4 is just a single chain. But now if we put the whole picture together, it turns out that class 1 MHC molecules bind to CD8, class 2 MHC molecules bind to CD4. So here's a target antigen presenting cell, here is the class 1 MHC molecule, here's the peptide, here's the T cell receptor, but what gives the specificity of the CD8 T cell to the class 1 MHC is the fact that CD8 sequences are homologous to sequences present in the alpha 3 domain of class 1 MHC, and that tells the T cell to only bind to a class 1 MHC molecule. In contrast, there are different sequences in class 2 MHC molecule, in this case the beta 2 region, which binds to the CD4 molecule, And this is what tells the CD4 T cell that its T cell receptor can only see MHC presented by class 2. And again, the way that I always teach students to remember it is the product is always equal to 8. 4 times 2 is 8, and 8 times 1 is 8. And that's how you know class 1 MHC is CD8, class 2 is CD4. Okay, and again, this also explains the structural motif of the CD8 and CD4 molecules. They're relatively tall in order to be able to bridge across the T cell receptor to see sequences in the MHC molecule that the T cell receptor is being bound to. Okay, any questions? Well, what are the functional implications of this specificity of class 1 and class 2 MHC molecules for CD4 versus CD8 cells? Well, as you all know, a CD4 cell is a helper cell. A CD8 cell is a killer cell. So if you are presenting antigen to a helper cell, you're going to get helped. If you're presenting antigen to a killer cell, what's going to happen to you? You get killed. So in fact, that's an important implication, I would think, if I was a cell. So here you have a macrophage. It's cruising along. It sees an extracellular antigen. As a macrophage, it just ingests it, it digests it, it presents a peptide from it. In this particular case, is this macrophage infected? If this is, say, a dead bacteria, it's ingested it, it's digested it, presented a peptide. Is it infected? No. So would you want to kill it? No. Or another scenario is maybe it is alive, but it's still inside the phagosome, so it's eminently digestible. So again, would you want to kill this macrophage? No. You want to save it. You want to recruit help. So therefore now you have CD4 T cell. For example, it'll make gamma interferon. It'll stimulate the phagolysosomes and now enable to be able to destroy the phagocytosed antigen and eliminate it. Or in the case of B cells, the B cell binds to extracellular antigen by virtue of the antibody specificity, ingests it, presents peptide and class 2 MHC molecule. Now it recruits T cell help, as I'll discuss in greater detail, to allow it to make antibody and class switch. Again, it's not infected. Would you want to kill it? No, not at all. However, let's say that you have a cell that's infected, and now, as I'll discuss again later, the peptides coming not from outside the body, but the peptides coming from inside the body. If you're making foreign peptides inside your cell, what does that mean? You're infected. And that means likely that you're not salvageable, that help us from a CD4 cell won't save you, and therefore, it's brutal to say, but the cytotoxic T cell says, in order to save other cells from being infected, we're going to have to sacrifice you to nip the infection in the bud, and then any foreign peptide seen in the context of class 1 MHC molecule by a CD8 cell, again, will basically stimulate lysis and killing of that target cell. Okay? So clearly there's very important distinctions and implications. Questions? So then, therefore, class 1 MHC molecules only present peptides derived from endogenous proteins. Proteins coming from inside the cell. The cell gets infected by virus. It encodes through the endoplasmic reticulum viral proteins being made. Now these proteins get digested into peptides and now get incorporated into MHC class 1 molecules, presented on the surface. Looking at a CD8 foreign T cell, CD8 cell sees it, recognizes a foreign antigen, and kills the cell. If these are self-peptides, they present self-peptides to the outside environment. Is that a problem? No, because there are no circulating CD8 T cells that recognize self-peptides, so therefore they're not going to kill the cell. Obviously, autoimmune disease is so devastating because if you do have T cells, CD8 T cells that recognize self-peptides, it's a disaster because then they'll start targeting all these cells expressing self, but when the immune system is working, it's not an issue. And therefore, cells expressing foreign peptide, class 1 MHC, CD8 cell sees it and kills the cell. But now for CD4, it's going to only be class 2 MHC by antigen-presenting cells, macrophage dendritic cells, and they only present peptides derived from exogenous peptides. So either proteins from outside or peptides that are derived from a phagolysosome because the class 2 MHC molecule comes into the phagolysosome, gets loaded by peptide, now gets expressed on the surface. B cells also have the ability to present in the context of class 2 MHC molecule, but B cells turn out to be rather selfish because in contrast to macrophages that will present any antigen they see, they just see it, they'll ingest it, they'll present it, B cells only internalize antigen if that antigen binds to its antibody molecule receptor. So if this B cell is specific, for example, for tuberculosis, and this is a soluble tuberculosis antigen it recognizes, it'll internalize it, digest it to peptides, and present it, because that recruits help onto itself to make antibodies against this pathogen. However, if this is like a tetanus or an albumin or another antigen that it doesn't recognize, it'll let it go by and will not internalize it. So it selectively only presents antigen derived from a protein that it itself recognizes. Whereas a macrophage will just ingest any antigen willy-nilly of whether or not it recognizes it. Okay, is that clear? And so therefore, CD4 T cells now come. In the case of the macrophage, it had been infected with TB, presents peptides from TB in class 2 MHC molecule. It's still within the vacuole, so it's definitely still salvageable. T cell sees the peptide, secretes gamma interferon, stimulates it to be a more potent phagocytic cell. In the case of a B cell ingests based on antigen receptor seeing it, presents peptide, and now activates its B cell to make antibody. And again, this is just to repeat, the B cell is presenting antigen from that it itself. So for example, if this would be HIV, for example, and this is an important concept. So let's say this B cell is GP120 specific. So you all know GP120 antibodies are critical in terms of neutralizing infection. So it sees the virus, and now it internalizes the entire, and this is, for example, is a dead virus, it internalizes the entire virus, chops it up, and now it presents peptides. It can present not only peptides from GP120, but it can present peptides from any portion of the virus. So it could be presenting peptides from P24, from RT, from integrase. As long as it gets it inside the B cell, it could enable peptides to be presented. The T cell has no idea what antigen the B cell recognizes. All it knows is that it's presenting a peptide that it's been pre-programmed to recognize. And I'll discuss this in a later lecture in terms of vaccine design, how we use this approach to allow us to make vaccines that against carbohydrates that normally would not be very antigenic. Okay, so now if we look in terms of where pathogens are located vis-a-vis where the MHC peptides will be presented, we basically have cytosolic pathogens. So for example, TB, once it leaves the vacuole, it's in the cytosol. Now it will be presented in class 1 MHC molecule because now once it's in the cytosol, the infection is not reversible, presented to effector T cells, and this cell is sentenced to death. If the pathogen is still inside the vesicle, so potentially you could still kill it by phagolysosome merging, now present to class 2 MHC, recruit CD4 T cell help, and will kill the intravesicular bacteria. And for B cells, internalizes it through antibody antigen binding and presents peptides, and then stimulates the B cells to make antibodies through MHC class 2 CD4 helper T cell interaction. Okay, any questions? Okay, now how if you were a virus and you would want to evade the immune system, what would be a great way of evading the immune system? Throw it out. Any suggestions?
Imitate the cell.
Imitate. Well, imitate the self, but it's hard for a virus to look like the self. Now let's think. How are we knowing that a cell is infected with a virus? What do we see? What is our immune system seeing? What molecules? Well, MHC2 is not necessarily saying the cell is infected. What's telling the immune system that the cell itself is infected? Right, and how are those viral proteins being presented? In what molecule? MHC class 1. Great. Now, if you were a virus and you want to stop the CD8s from knowing that it's been infected, what molecule would you downregulate?
The same molecule.
the MHC class 1. So it's in a sense, if someone kidnaps you, the first thing they do is they put a gag over your mouth so you can't scream for help. So viruses do exactly the same thing. And it actually turns out, and this is a paper that Bruce Walker was co-author on a number of years ago, showing that HIV NEF actually downregulates class 1 MHC expression, prevents it from being expressed on the surface, and therefore prevents the cell from being killed by cytotoxic T cells. It's a brilliant approach that viruses have used. Does that make sense? They're really creative. Now, of course, we're not stupid. We know that viruses would try to do something sneaky like that. So therefore, we've come up with a way of dealing with that. And that way is by using another kind of cell called NK cells. And NK cells are natural killer cells. So are these helper cells? No. Again, immunologists are your friends. It's telling you that they're going to be doing some good killing. And in fact, these were really actually originally described more for cancer immunology because cancer cells downregulate class 1 expression. It turns out that NK cells have the ability to determine whether a cell is expressing class 1 MHC or not. If the cell is not expressing class 1 MHC, then NK cells are designed to kill it. So the way it works is that the NK cells have two receptors on its surface. One is called an inhibitory receptor that binds to MHC class 1 molecules. If it sees MHC class 1, it sends a negative signal to the NK cell that tells this other receptor called the activating receptor not to signal the cell to die. All cells turn out express molecules on their surface that can interact with this activating receptor. However, if there's no MHC molecule on the surface or it's been changed, now this inhibitory receptor has nothing to bind to. Now this activating receptor has nothing stopping it. There's like nothing turning on the brakes, preventing it from killing. And now what happens is that this activating receptor is unopposed. It now sends a signal through this cellular ligand and tells the cell, time for you to undergo apoptotic death, and then it kills the cell. So therefore, potentially a way that our immune system uses to circumvent immune evasion by downregulating class 1 MHC molecules is by saying, I know all cells should express class 1 MHC molecules. If I don't see a cell expressing class 1 MHC molecules, something fishy is going on. Therefore, it's probably because it's been infected. Now I need to kill the cell. Does that make sense? And again, this is actually very important in tumor immunity because it's very frequent that when tumor cells downregulate class 1 MHC to prevent cancer-derived antigens from being presented, and NK cells, again, play a critical role in killing tumor cells. Unfortunately, HIV is also relatively shrewd, so it's come up with another way of, therefore, avoiding NK cell killing. And it turns out that NEF also downregulates that activating receptor, this activating receptor that must be expressed by the cell in order to provide the death signal. And in downregulating this also blunts the ability of NK cells to kill HIV-infected cells. So again, this is another example of how we have a very well-developed way of circumventing pathogen evasion of the immune system, but HIV is really amazing in terms of its ability to come up with new ways of evading the immune system. Okay, is that? Okay, so therefore, really to end up with, the questions I started talking about were how do B cells and T cells see antigen? The answer is they see it differently. B cells see three-dimensional structures, whereas T cells basically see peptide. So the three-dimensional tertiary structure is irrelevant to how a T cell sees antigen because it's eventually only seeing peptide. How do T cells distinguish between whether a foreign antigen is being presented to recruit help or because the cell is infected? And the way it distinguishes is based on what MHC molecule is presenting it. If it's being presented by a class 2 MHC molecule, that implies the protein came from outside, so the cell itself is not infected, or it came from a vacuole and the cell can still be saved. However, if it's presented by class 1 MHC, that means it must have come from inside the cell, either made by a virus or in a cytosol, and therefore the cell needs to be killed. And the class 2 MHC, and why do some T cells kill and others provide help? Because CD8 T cells see class 1 MHC, which tells that the cell is infected and can't be saved, whereas CD4 cells see class 2 MHC and means that it still can be assisted. And therefore, that kind of puts it in context. And then we have a half-hour break. And then in the next lecture now, I'll do the second part, discussing exactly how MHC presents peptides and the structural elements that are involved in that process. Again, thanks a lot for your attention.